Greetings, everyone. I'm Amanda Miller, Associate Director for the National Consortium for Data Science, also known as NCDS. It is my pleasure to welcome all of you to our August 2023 DataBytes event with Dr. Kimberly Robasky. This event is titled AI in Target Identification and Drug Discovery, Transforming the Future of Medicine. Before I introduce our guest, I'd like to remind you that the goal of DataBytes, this speaker series, is to share developments or new perspectives in the field of data science. These events often highlight work happening in industry to provide a pathway for us to connect industry with academia. We design NCDS events to build community. However, we also respect the limited time of our speakers and panelists, and we ask that attendees refrain from posting comments and questions in the Q&A until Dr. Robasky completes her presentation. There will be a moderated Q&A toward the end of our time together. I'll add that we would love to continue this conversation or any ideas sparked today in our listserv. You can direct message my colleague, Jayshree Jagannatha, and we'll add you to the community. With the housekeeping behind us, I am excited to welcome our speaker, Dr. Kimberly Robasky. Dr. Robasky is currently the Associate Director of Machine Learning and Artificial Intelligence at Arrakis Therapeutics. In this role, she manages a team of scientists focused on applying advanced data science and machine learning tools with the goal of extracting and integrating insights from various types of internal chemical and biological data sets to drive innovative data science and deliver novel therapeutics to patients. She's also worked as the head of translational science at the Renaissance Computing Institute at the University of North Carolina at Chapel Hill, amongst others. And I'm not sure which is more challenging, managing scientists or data sets, but I think I know the answer. So without further delay, I hope you'll join me in welcoming Dr. Kimberly Robasky. Kimberly.
Thank you so much for that kind introduction. I appreciate that. And thank you everybody for joining me today. I'm looking forward to talking to you about this interesting topic that's near and dear to my heart. Today we have a nice mix of people. I see about a quarter of you have not had hands-on experience with ChatGPT yet, and about 3/4 of you are working with machine learning in your company, and maybe about half of you are doing machine learning at work, and only about 1/3 of you have experience professionally in drug development. So I'm going to try to present to you, make sure that everybody can follow along based on where you're coming from. But first, I want to level set on the definition of AI. A lot of people think that I've spoken to, believe that machine learning is not part of AI. And so I just want to level set the definition for today. We're going to be talking about machine learning as it is part of AI because machine learning is the underpinning of a lot of what I do for supporting our AI strategies. And so we're going to be talking about that as we talk about the implementation. Okay, so based on this experience, I want to talk to you guys about the data origins for target identification and drug discovery. And I'm going to give you an overview of the drug development process based on the knowledge that about 2/3 of you have not had that professional experience. Then we're going to move into the potential impact that AI has on target identification and drug discovery. And finally, we'll move into actual use cases that I've been involved with for accelerating target identification and drug discovery with AI. We'll keep it high level, but we'll also get nicely into the weeds so you have a really good, solid understanding with real applications, and we'll end with takeaways. So starting with the data origins, I want you to really understand the foundations of what is making this AI, what's driving AI in drug discovery and target identification. But we're going to start with talking about the drug development process so we understand how all of this fits together. And the process begins with target identification, moves into the drug discovery for those targets. From there, we go to preclinical testing in cells. Then we move to testing. We introduce humans for the first time in the testing in clinical trials. And the whole thing culminates with an application to the FDA for new drug approval. And so in target identification, we're looking at targets like genes, proteins, RNA molecules. These are the things that are targeting that we can use for a potential drug to target a disease. From there, we move to drug discovery. Drug discovery is where we try to modulate the target in order to treat the disease. And this entails often things like computer models to identify the drug candidates and then screening of those drug candidates using libraries of thousands of compounds against your target experimentally. From there, we move into cells with preclinical testing, where we evaluate the pharmacological activity in disease models to assess the safety and the toxicology of the drugs before, before moving into humans in clinical trials. In clinical trials, we usually have 3 phases. The first phase is for safety. You've got maybe 100 humans, takes maybe several weeks to a year, several months to a year. Is in phase 2, where we're looking at efficacy and dose range, and then we maybe have 1,000 humans, and it could take up to 2 years. We move into phase 3, that's your pivotal trial, and here we're looking at efficacy in larger populations with thousands of humans, and that can take several years. From there, if your drug makes it all the way through all these different processes, then you can now go for a new drug approval and submit to the FDA your drug label. So your drug label is what you're trying to get at with all of these different steps in the process. That label is going to tell you what are the drug indications, what kinds of diseases you can use this for, what your dosages are, what populations are appropriate, and so forth, and the potential side effects. And so that's all spelled on the drug label. That's what goes to the FDA for new drug approval. It takes 10 to 15 years to get there, around 10 to 15 years. And you only have a success rate of about 10%. Arguably, people say around 10% starting from phase 1. And even after you've gone through all that and all that time and expense, your drug can still fail in post-market approval surveillance. So there's a lot that you go through here. And the part that we're interested in for this conversation is the target identification and the drug discovery. So the first part of this whole thing. And my company, Arrakis Therapeutics, we talk about, we actually focus on targeting the RNA molecules. Our platform, many platforms look at the proteins, we look at the RNA. At Arrakis, we focus on the interface between the small molecules, RNA binding small molecules, and genetic medicine. And more explicitly, we look at the RNA because there's about over 500 drugged proteins. And we think there's about today, a few thousand druggable proteins, but there's 20,000 coding genes, and there's over 200,000 RNA transcripts. And so Arrakis and a few other companies are looking to reap the benefit of that additional capability for drugging the genome through the RNA transcripts. And we think we will be successful. We think this is a good platform because there are already a handful of drugs out there that do target the RNA molecules and do so successfully. And so the way we're doing this is by modulating RNA targets, changing the gene expression, modulating them through the translation, the splicing, and the degradation of these RNA transcripts. And so for the translation aspect, we're looking at the 5' UTR region upstream of the RNA molecule, where that area that's responsible for controlling translation into a protein. The splicing area is the introns and the exons within the RNA molecule that controls how the RNA molecule gets sliced and diced after prior to that translation. And then the degradation is we're looking at that where we can actually recruit RNA degraders to, again, modulate or downgrade these RNA transcripts. So that's kind of the focus of what we're going at and how we're doing the approaching this. And our platform looks a bit like this. We have target identification, selection, screening, and target engagement. That's sort of the 3 steps that we go through. This is generalizable to a lot of biotechs, including, for example, protein targeting companies. And so this slide here is your roadmap for the next 3 slides, where I'm going to tell you a little bit more about each one of these 3 steps. So in the target identification and selection, we're selecting our targets. We start first by looking at the one-dimensional sequence over here. Can you see my prompt okay? Let me know if you cannot. We start with this one-dimensional sequence on the left, looking for the transcription start sites, and then we move into folding that one-dimensional sequence into 2 dimensions in silico on the computer using computer models to determine whether or not there are interesting bulges where a compound could bind, and then we experimentally validate the 3-dimensional structure. And there's a lot of other things that go into this in assays and things that we use for that target identification selection, but that gives you the idea of it. And from there, we go into screening. We have the target that we're interested in. Now we have a pooled library of small molecules, thousands of small molecules, excuse me, that we will assay against that RNA target, which will result in a number of RNA small molecule complexes where those compounds will bind to the RNA molecule. And then we use size exclusion chromatography, or SEC, to separate out those compounds from the targets. And from there, we can use mass spectrometry to determine which one of those small molecules actually bound to the target. So that's SEC mass spec. So SEC mass spec is the first step in our experimental screening of our targets to find RNA binding compounds. And there's other assays that we use further downstream to sort of refine that, but that's SEC mass spec is the first thing. But the other side of it is, with the SEC mass spec, you have one target and thousands of compounds, but we also need to be able to see if a target can bind more than one, if a compound can bind more than one target, because we are interested in understanding off-target effects. We really want the compound to only bind one RNA molecule, one target. And so we have this technology called PearlSeq, and you can Google that. We've published this platform. And what you have here is you start with your target, and it may have several different potential binding sites, but we have a compound that we adhere a warhead to. We call it a warhead, and when you use photolysis, it cross-links your compound to the target using that warhead, so that you can do further evaluation of those complexes. And this allows us, instead of looking at one target for many compounds, we can look at many targets for 100 or so compounds instead. And so that allows us to see if a compound is binding to more than one target, or if it's being selective as we would like it to be. But it also allows us to understand the target occupancy in cells, which is really important, because if this compound can't actually bind to the target in cells, that it could be competing with other things in that cell, and we want to make sure that we understand that. So these are all the different assays that can generate data that can help us to understand this interface between the potential drug candidate and the target, that what they call ligandability, that ligandability interface. And so here I wanted to sort of show you a little bit more about, what the data look like that you get out of this. For our company, we have a data aggregate we call mineable data assets, or MIDAS. You're going to hear me mention MIDAS again as we go along. MIDAS, we have now to date over 180 targets that we've screened, and now we have binding data for about 30,000 compounds. We actually have binding data for many, many, many more compounds, but this is the ones where we actually had some SEC mass spec gave some positive binding data. So we have the negative controls as well. And this compares favorably to other data sets in the public domain, such as RSIM, RBIND, and Robin. But I'm comparing them here, but they're not exactly comparable. But if you wanted to look into those other data sets, you get kind of a sense for what kind of data we're working with when we talk about the MIDAS data at Arrakis. It gives us lots of binding information about these compounds and these targets. All right, so now I hope you have a good idea about where we fit into the whole drug development spectrum and where the data comes from. And I just want to talk a little bit more about what you actually have to do to make these data machine learning ready, because boy, there's a lot of work you have to do. This is about 80 to 90% of the effort that goes into modeling these data is just in getting the data ready. So you have to prepare the data, make sure that you understand the quality of it, identify any biases, because that can really mess up your modeling, find any missing values, identify errors, outliers, and so forth. And then you want to really visually explore the data well so that you can engage your stakeholders to better understand what kind of harmonization you need to do, how you might actually correct any biases or correct any errors and so forth. And I just want to say, when you move into cleaning and verifying that data, that the harmonization and the encoding is also probably, like, expect that to be the biggest amount of effort. Sometimes you get lucky and you have very well curated and collected data, but it's extremely rare in my experience, particularly with healthcare data. Okay, so you've done all this engineering, 80 to 90% of your effort, but you still can't jump into modeling yet. The first thing you want to do is a utility assessment. And in the utility assessment, what you're doing is you're just creating a very rough baseline model to ensure that your data is actually producing signal for the question that you're trying to answer. So if you create a model and it does no better than random, maybe you don't have the right data set. So that's your utility assessment. And once you've shown, yeah, you can actually do better than random in your modeling, then you want to define your modeling goals, performance, how much compute you have to use on it, and so forth. And then you want to conduct and record your modeling experiments that you're going to do to decide what hyperparameters to use, what kind of model to use, what architecture, and even what type of approach, what kind of algorithm. I'll tell you, the last thing you ever want to do is a neural network. If you can use any other kind of model, always use a different model. Your neural net is the last thing you want to do. Good rule of thumb. And then once you have that figured out, then finally you can deploy your model and monitor it for performance and drift. Okay, so that's a lot of effort, as you can see. And you might be asking yourself, gee, you have to do all this to get all this data and go through and make it modelable. Is it worth it? Well, why would we want to do this? Let me tell you, the reason is because the target identification drug discovery is extremely expensive. It's extremely costly. It's a long slog. You're going to spend $2.5 billion to go from a target to an FDA drug label. Arguably, some people say more, some people say less, but billions of dollars is what you're going to spend. And the chances of your success getting a drug that's actually going to help your patients is slightly under 10%, starting from phase 1. That's after you've done the target ID and drug discovery. So, this can take years. It's expensive. Why do we think AI is going to make it better? Let's look at some of the significant events that have happened over the past 5 years. In 2019, in silico medicine, coming out of the University of Toronto, went from target to drug candidate in just 46 days using their AI-based screening methodologies. And then the year after that, we got our first AI-designed drug entering trials. And a couple of years ago, big, huge breakthrough. You probably read about it. AlphaFold was able to predict all the protein structures in the human genome. Incredible. And then just last year in silico medicine again brought us our first AI discovered drug and AI designed target into trials. And then this year, generative AI being the thing behind ChatGPT, you guys have all heard about it in the news, brought us validated and created de novo antibodies from generative AI. And this year, which, one of the most exciting things I think over the past 5 years is that the FDA actually granted orphan drug designation to an AI discovered and designed drug, again, in silico medicine. So, the proof is in the pudding. There's a lot of really big things happening with AI in this field. And so let's talk about the overall opportunity then for target identification and drug discovery part of the drug development process. And here I want to mention large language models, these LLMs that, again, have been all over the news. If you watch CNBC, lots of startups, it's accelerating the rate at which we can transform data into research, the rate at which we can understand and get valuable information out of our data. And you're seeing deep learning is enhancing our ability to do the target identification, making it more efficient, more effective. These generative models are enhancing the creativity behind lots of things, right? Behind creating images, behind language, the language models, music even, and now drug designs and chemical structure optimization. And now we can start combining the target identification pipelines with the drug discovery pipelines to understand the biology behind the ligandability, this interface between the target and the compound. So the AI can predict that. And we can also use the AI for virtual screening and molecular docking to help us find lead compounds and optimize them faster. So all of this opportunity is there. And on top of that, we have new opportunities for sharing and reusing trained elements. And so, I work at a commercial company, we have IP, we don't want to share our data, our special sauce, but we can share our embeddings, for example. The embeddings are the learned relationships between the residues, the amino acids and nucleotides of the genomic sequences. We can share our foundation models, which is just basically a matrix of weights for our neural networks. And so these are things that you're starting to see happening in the field. So let me talk a little bit more about leveraging AI for target identification. Specifically, the opportunities for target identification include mining our own private target knowledge bases. So every company like mine has a private knowledge base. A lot of it is unstructured data. And now we can use these LLM foundation models, like what drives ChatGPT, to fine tune those on our own data to help us accelerate our understanding. We can now combine multimodal data like we never could before, using things like transfer learning. Traditionally, everybody knows you can't combine expression microarrays with RNA sequencing data, but now you can, because you can train a model on the gene expression microarrays data and then transfer learn that to RNA sequencing data that you can then train and make predictions on using RNA sequencing data. And that's just one example. Also, this predictive modeling with these techniques are allowing us to have more data-driven approaches to where to go next for experiments. And we can even extend existing areas of research that are really advancing quickly, things like explainable AI, ethical and safe AI, and calibration. With explainable AI, we can look at, for example, the ligandability predictions. That is, if you have a predictor of whether this compound and this target are going to bind to each other, you can use explainable AI technologies like LIME and the Shapley libraries to determine which residues and which structures were actually responsible for that ligandability. And we can use these kinds of in silico predictions to reduce our costs, to look across multiple populations, to make for ethical and safe use of AI. And regarding calibration, or this field called conformal inference, we can use this to identify in a data-driven way thresholds instead of arbitrarily picking thresholds. And we can compute confidence levels so that we can reduce the costs in our and better control costs in our target predictions. Okay, so from here I want to talk about two use cases. One is these are both in generative modeling used in target identification. Use case one is around using OpenAI's GPT-4 for general target research, and use case two is for parsing structured data from unstructured data. I'm going to talk about PubMed abstracts and how can we read those faster. But for use case one, since some of you may not have used ChatGPT yet, I want to show you on the left side here is what a conversation with ChatGPT might look like. And here I'm asking just simply ensemble ID for CFTR. CFTR is a famous cystic fibrosis gene. I don't even have a full sentence here. I'm just saying ensemble ID for CFTR. And GPT-4 says, nope, I don't know what the ensemble ID is, which is terrific because the older models will hallucinate and just make up an ensemble ID, and then you have to go and check and see if it's real. So the older models have a real problem with these hallucinations, particularly for citations and URLs. So you always have to be very careful checking those. So this is nice. GPT-4, the newest model, tells us, I don't know what that is. And then I asked it, well, what chromosome is it on? And predictably, it answers it's on chromosome 7. That's great. That's right. And then I just ask it diseases. I don't even say what diseases. I say just diseases. It knows what I'm talking about. It remembers the conversation. And of course, it tells me cystic fibrosis, but then it tells me 3 other diseases that I did not know were associated with SNPs and mutations in the CFTR gene. And sure enough, if you go and you Google them, yeah, there is actually evidence behind that relationship. So this is really, I hope this demonstrates the opportunity here for using these kinds of LLMs to really do some good digging on target identification. The second use case I want to show you is how you can turn unstructured data like abstracts or PowerPoint presentations or PDFs into structured data, which is basic tables that you can then use for your machine learning. So in PubMed, you can download about 22 million abstracts from NCBI. And if you filter those for any that just mention gene symbols and accession IDs, now you're down to 14 million. And if you just look since 2015, you still have 5.5 million abstracts. Well, the older GPT-3.5 model from OpenAI can parse those at about 1.5 seconds per abstract for 1/10 of a penny each. And so if you want to read all 5.5 million abstracts, well, you're still going to pay, you're going to pay $7,000 or 100 CPU days. And if that's just too much expense, you can pre-filter these abstracts even more aggressively. You can work on refining the prompt, making it smaller, more effective. And you can even now fine-tune the model by feeding it new data and getting it better at producing the structured data that you want, so you don't have to incur quite as much of an expense. And so this is what the data would look like, right? So what I did was I took 4 abstracts that I knew that the MYC gene were in, and I expected the model to be able to read the abstract and find the MYC gene. And the columns in the red box on the left are the columns that the model generated. These are generated from the abstracts. And sure enough, it did find the MYC genes, and it found also what diseases the abstract was talking about, if it was talking about a disease, and what the target types are. The code for this is super simple. It's just a couple of lines, but you have to put in a few more lines in order to handle exceptions, because the model can, as I said, it can hallucinate, it can time out, it can do funky little things. So you have to catch those and filter them. But more of an issue is the prompting. The prompting aspect on the right here is super tricky. And it's not just a matter of learning the English language and knowing how to speak English. You actually have to understand how these models will parse your language and how to get it to give you the information in the format you want, how to get it to not hallucinate, how to be succinct enough so that you're not spending too much on the prompt. So the prompting is tricky. You might have heard about this new field called prompt engineering. It's a real thing, and I would encourage you to develop those skills either in yourself or in your organization. Okay, so how do we accelerate target identification now? By leveraging AI for lead compound discovery. So we talked about the target identification side. Now I want to talk about the compound discovery, the drug discovery side. And the opportunities on the drug discovery side include, as I already mentioned, in silico screening, in silico medicine at University of Toronto is doing amazing things with their in silico screening platform. But also we can use machine learning to predict which compounds we want to prioritize based on their molecular properties, the pharmacokinetic safety profiles, and so forth, drug properties. And so now I'd like to talk to you about a couple of use cases for supervised and unsupervised learning, where we predict the compound screening outcomes. And I'm going to also talk about using unsupervised learning and drug discovery. And from here, then I'll we'll go into lessons learned and QA. Okay, so the first use case, again, mentioned MIDAS before, the mineable data assets, right? So here we're using MIDAS again, and we're trying to predict those screening outcomes. And specifically, we're talking about the SEC mass spec that I mentioned earlier, size exclusion chromatography. Can we predict those outcomes? The SEC mass spec gives us a score for each compound target binding, how strongly it thinks it binds it, with 100 being maximal binding and 0 being no binding at all. And what you're looking at here is along the x-axis, along the bottom here, starting on the left, if we set a score threshold of 90, then what we see is we get, if we look over here to the right, the secondary y-axis tells us we have about 2,000 observations. And so 2,000 observations in a balanced data set, we have 1,000 observations that have a score in SEC mass spec of 90 or greater, and 1,000 observations that had 0. And we have a lot more than 1,000 observations, but we, subsample in order to select a balanced data set. And then we create a baseline model. Remember, I talked about utility assessment of your data. So we create a baseline, just a simple model, to see if we can do better than random. And in this kind of a binary predictor, random, you would see a performance of what they call PRC. AUC is one of the predictors a performance measure of your model. You want to do better than 0.5. And what we're seeing when we set the threshold at 90 is, we're getting much better than 0.5. We're getting over 0.65, which is terrific. And in fact, if we keep lowering the threshold for our training data set expands. We get all the way down to 45. Now our training data set starts to level off, and we see here predictably that our performance of our model continues to go up. And then at about 45, when we're not adding as much data, it kind of levels off. So this is great. These results are great because what this is showing is that you can actually predict which compounds are going to bind in RNA. And if you can do that, then you can look at the millions of compounds that are commercially available and figure out which ones are going to be your RNA binders, which ones are worth looking into further. And it's a little bit more complicated than that, but that kind of gives us a little bit of an idea of where to look. Okay, so that was use case 1. And then for use case 2, we're going to try and answer the question of, do molecular fingerprints of RNA binders tend to cluster together? That's a mouthful, right? So first of all, what's a molecular fingerprint? So a molecule is essentially a graph. It's very hard to train a neural network effectively using graphs, but you can, chemists have this trick where they kind of flatten it down into a vector called a fingerprint. It's a regularized vector. And furthermore, you can take those vectors and do math on them to figure out which compounds are structurally similar to each other. And so what's really nice is if you have a bunch of compounds that are structurally similar to each other, and also they bind RNA, well, then new compounds that you could potentially purchase, if you can find them that are also close fingerprints, then maybe they would also bind RNA. So the question is, do the fingerprints of the binders tend to cluster together? We don't know. And so what we did was we took 3,000 compounds from MIDAS, measured their orthogonally confirmed dose-dependent binding. What I mean to say here is that, okay, so I've talked about the SEC mass spec, but that's not the end of the line. We then go on to orthogonally confirm that binding. And so now in this experiment, we're looking at the orthogonally confirmed binders. These are really binders. And then we look at the ones that didn't confirm. And we computed the fingerprints on all 7,000 of these compounds, computed the distance between all of those fingerprints. So we have pairwise distances everywhere. We have an adjacency matrix. We have this big, fully connected graph. It looks like a hairball. And then we clustered that graph to find bits that, that were very similar based on their Tanimoto distance. And then we determined how many clusters were in that graph, which was 3. There were 3 clusters. And then we visualized that graph, because it's just a hairball now, by unrolling it into 2 dimensions using UMAP. And once we have that, we recolored it red or blue based on whether the compounds confirmed for RNA binders or not, so that we could see if there was something about the structure that was causing them to bind. So this is what the graph looks like, the UMAP. After we cluster it, we color it for the clusters. we found 3 clusters, and here we see a nice cluster, that cluster 0, all these compounds are very structurally similar. Cluster 2, all these compounds are structurally similar. And then there's this big sort of green smudge. These are, well, more similar to each other than they are to 0 or 2. They look like there's little subclusters, but remember, this is kind of a hairball, and this is artificially projected into 2 dimensions. That's what the clusters look like. They have nothing to do with binding. So what do you think is going to happen if we recolor this now for those compounds? Remember, each dot is a compound, but if we recolor it for those compounds that are actually binders, well, we really, really hope that we find the clusters 2 and cluster 0 will be the same color after we color it for binding, because that'll tell us that the structure is important. And sure enough, that's what we find. So in cluster 2, these are all compounds that not only are structurally similar, but look, they're the ones that are blue means that they also confirmed in binding. And so there's some red ones in there, but mostly they're blue. And in cluster 0, mostly they're not binders. And so if you have a new compound and it's structurally similar to somebody in this cluster, then probably you have a better chance with a compound that's structurally similar to this cluster. That's kind of what we found with that. So it's just another piece in the puzzle. And so for our takeaways, I hope that you can understand that AI is never going to replace experimental validation, but rather it's going to help drive us where to look for the experimental validation, so that when we integrate AI and experimental validation, this is going to really help us accelerate safely and successfully to produce more robust, more reliable results, and to facilitate data-driven decision-making, as opposed to sort of arbitrarily making choices that are a little bit more subjective on where to look next. And we can continue to improve using the iterative feedback from the experimental results. I hope that I've convinced you that AI has a transformative potential in target identification, drug discovery. And as we move forward in research, collaboration between the AI experts, biologists, chemists, and clinicians are going to really drive the innovation in the field, those 4 groups of people. Thank you for your time. I'll take questions.
Great job. Thank you so much, Kimberly. That was fantastic. So folks, now we do invite you to engage with Dr. Robasky and some of this material that she's presented today. So just go ahead. If you have comments, feel free to click on the Q&A button at the bottom of your screen and type in your idea or comment, and go ahead and you are welcome to submit anonymously if that's preferable for you. And when you're finished, just hit the blue box with a thin white check mark. So we will go ahead and begin with the Q&A. And I'm going to give folks a couple of minutes to type in any questions that they might have as well. All right, so looks like we have our first question here. Dr. Robasky, do you see that on the screen?
I do.
Great. So you might want to, I'm going to invite you to answer live. And if you wouldn't mind reading these questions as you answer them, that would be great.
So I'll start with the second one, and so the first one says, I am currently doing a PhD in nanoengineering. I have questions about AI and molecular modeling for catalysis. And if there's a specific question there, I'll invite you to go ahead and ask that, and I'll do my best. I don't have a PhD in nanoengineering, so I don't know if I'll be able to answer your questions, but I'll do my best to help you with that. But the other question is, have you identified new targets based on the trained model you learned from 10,000 targets tested in SEC mass spec. Well, I will tell you that this model that I'm presenting today is very much hot off the presses. And as I mentioned, the results that I just showed you are based on a utility assessment of the data set. And so this particular model is just a baseline model. And so you wouldn't really want to use a baseline model for driving decisions. In fact, the next step would be to then work with stakeholders to identify performance benchmarks and then refine the architecture of the model and the hyperparameters and so on and so forth. And then you have a model that you can deploy into production. And it's important to deploy it like that so that you have results that are reproducible, that enables interrogation and allows you to fold in that explainable AI, those different techniques I mentioned before. So this is just a baseline model, which might be very unsatisfying to your question, but I will tell you that this is only one of many of the techniques that we're using in AI and machine learning, and there are other models that we have been using on different data that we have been selecting compounds from to expand our compound library, and rather successfully. And if you go to our website, you'll see that we have a couple of things in the pipeline right now. So even though this particular model that I showed you today is sort of really, cutting edge, out in front and not really used in production right now, we do have a number of machine learning techniques that are that we are using in production. Does AI also give drug effects on the target? So I think the question here is asking about, and I'll invite you to clarify that if I'm not answering the right question. But I think what you're asking about is sort of the, there's a couple of things. There's 5 rules that make a compound drug-like. Or should it be validated? Okay, always validate with experimental results. Always, always, always. Really, really important. AI cannot do it all. AI can just help you. You have this enormous universe of 200,000 transcripts, for example. So which ones should you look at first? You want to look at all of them, but which ones should you look at first? And AI can kind of help you with determining that alongside of other experimental results and other models and all kinds of things. It's really, there's a lot that goes into trying to figure out what do we look at first. But once you figure that, okay, we think we should look here first, you always need to do the experimental validation. But looking at the drug effects, as I was saying, there's these 5 rules for determining if a compound has drug-like properties, and that's just, I think, for the most part, AI is not a big help on that, but understanding off-target effects, understanding the biology behind the interface, the ligandability of the compound to the target, and understanding the in-cell, what's going to happen in-cell. This is where AI can really help, because you can, as I mentioned on one of my slides, do molecular docking in silico so that you can, before you run your experiments, which are really, really expensive, you can make some predictions on what you think is going on there so that you can make the best use of your dollars for which experiments to run to really confirm that ligandability interface and get more lessons learned using these models. Does that answer the question? I hope that's kind of what you were driving at. Great.
Great. Thank you. Thanks for those questions that folks have been sending in. Do feel free to continue to post questions or any comments that you might have as well. We're happy to answer those. And Kimberly, just, since we have a bit of a quiet audience today, if you want to talk a little bit more about some of the other ways that machine learning is being used at Arrakis, or, we've got a pretty scientific audience that's in attendance today. If you want to talk about any other applications or any other platforms that you're finding that are pretty compelling for our data science folks that are in the room, so to speak. I would love to hear a little bit more about that, as folks maybe get the courage to type in maybe a final question before we wrap up.
Yeah, these questions are great, and there's no bad questions. So I thank you. I really appreciate it, and I love talking to you guys. Yeah, so I think there's a tremendous amount of opportunity for AI, especially on the target identification side, particularly with the advancement of these large language models. A lot of the techniques that we are seeing applied to understanding human speech can also be applied to understanding the genome, because the real problem with applying machine learning, I think, to the human genome is that the sequence matters, right? The sequence matters. And so now that we can develop these models that talk like humans because they can understand the language, the sequence of the language, the order, we can do the same modeling on the nucleotides in the genes. And so there's a tremendous opportunity here, I think. And we're starting to see those models come out now on the target side. On the compound side, I mean, computational chemists have been using machine learning for quite a long while, and we're already seeing really great models of using graph neural networks to understand the compounds in their structure, and how that and how to find compounds that are going to ligand to your proteins, how to molecular dock, all that sort of, all those models are coming out of the computational chemistry side. So there's really been a lot of advancement there. And on the target side, on the biology side, I think there's a lot of opportunity. So if you're looking for a place to sort of dig in and really make a mark, I think there's a lot that can be done there. It's very interesting. There's a few experts in the field that are already publishing these models and making leaps and bounds in there. But I think there's a lot of opportunity, especially in the RNA biology, where I work, in understanding what the structures look like for these RNA transcripts, because protein is much more stable and structured, and you've got them all folded, and this is what the structure looks like. But RNA is much more floppy, right? An RNA molecule is like this, and so finding a stable structure in an RNA molecule is a bit harder, and there's a lot more open field there. And so I think there's a lot of opportunity for machine learning there and AI. Thank you so much for the great talk. Thank you. And for demystifying a challenging topic. Yeah, it is challenging. Could you comment on what you think future directions are for this field? Oh, okay. Well, I was just doing that a little bit. I think, like I said, I think that the target identification, both on, I'm more on the RNA side, but also on the protein side, I think that's going to be really important. And I think there's going to be a lot of opportunity here for the foundation models. I really want to encourage people, especially if you're in academia, I think we have a large academic audience here, into looking into and providing foundation models. One of the things that I would love to see is a foundation model built on those public data sets, for example, that I just provided. And there's a really good, there's a terrific protein data set, ligandability of proteins out there. And if somebody built a ligandability model, foundation model on that, then I can see companies taking that foundation model and transfer learning it to their corporate private databases, and you could see a real advancement in in drug discovery. I can also imagine a lot more being done with conformal inference. So I don't see a lot straight across the board in machine learning and providing these confidence intervals in the predictions. And so, if you're in Bayesian statistics, you're going to be talking about this, but if you're in neural networks, I don't see a lot there. And so I think looking into, that's the term of art is conformal inference, but really we're talking about is calibrating these models so that you can have data-driven thresholds. I hear people all the time, how do I pick the threshold? If you can calibrate the model so that you can pick the threshold, and also that same approach can allow you to specify confidence intervals on your predictions, that can do a lot for controlling the costs in healthcare. And so I would encourage you, if you're an academic, look into that too and see, what papers, what libraries can you publish? If you want to write some Python, you can do that. I'd use your library. Yeah.
All right. Well, thanks so much for your responses there. I just want to confirm. Oh.
Oh, I want to answer this because yes,
Okay.
please. We have a very strong internship program. In fact, my company is based in Boston, and so we work with, my company has great co-op opportunities. Arrakis Therapeutics is the name of the company. Look and see with your university if your university offers a co-op. If it doesn't, reach out to me, because we're just now starting to do the recruiting, and I can work with my HR to, yes, please connect with me on LinkedIn. I can work with my HR representative to make sure that your university has a cooperation with our company for this. In fact, my company's in Boston. I'm located in North Carolina. I'm an adjunct to UNC Chapel Hill. And I also encourage them, they're going to have a relationship now with NC State, because, for the first time, so that we can take advantage of some of the engineering coming out of NC State. And like I said, if you're at a university, NC-A&T, okay, yeah, reach out to, I don't know, I'll find out if NC-A&T is on there, but you can apply. I will say that the internships, the co-ops at Arrakis are terrific. I mean, they're really fantastic. You will do a lot. You might even get a publication out of it. They're very competitive. Just warning you, we've worked with dozens of schools around the country. And for the co-op that I'm going to be looking for, I'm looking for strong engineering skills and maybe some machine learning. And so if you have that, please, by all means, reach out to me on LinkedIn.
Thank you so much, Kimberly. This was fantastic. And thanks to all of you who have joined today and stuck around. Of course, to our wonderful folks who have asked a couple of questions. We really appreciate those. I want to invite all of you now to complete an evaluation form for this presentation today, for this webinar. We would love to hear back from you about the kind of topics you're interested in learning more about, and the kind of speakers that you'd like to hear from as well. So please do take a moment to provide that feedback via the link that was just dropped in the Zoom chat. We are also always looking for DataBytes speakers. For those of you interested in learning more about our programs in general, please visit datascienceconsortium.org for more information about our upcoming events and the other types of work that we do. And with that, I would like to wish you all a fantastic rest of the day. And huge thank you so much, Kimberly. This was so great. Really appreciate your time and your knowledge and your wisdom and all of those things you've graciously provided today. So thank you.
Thank you for the opportunity.
All right. Take care, everyone.